Greetings, ladies and gentlemen. I hope everyone is staying safe and staying home in these unprecedented times. I'm Rahagir Salekeen from Kuala University, Bangladesh, and today I'll be presenting a portion of my undergraduate senior year thesis, which will be focusing on Heritiera fomes extracted phytocompounds and their roles in inflammation. So Heritiera fomes is a mangrove plant native to the coastal regions of India and Bangladesh, and it has to withstand an enormous amount of biotic and abiotic stresses in the harsh saline ecosystem. And how it performs this is by producing a plethora of xenohormetic compounds or phytocompounds that helps it fight oxidation, salinity, and microbial attack. The reason I'm mentioning this is because Heritiera fomes has been endogenously used as an ethnomedicine for centuries, and recent advances in phytochemical analysis have revealed that these same compounds that help Heritiera fomes fight off abiotic stresses are the same ones that produce therapeutic effects in humans. So our research lab has long been exploring the anti-inflammatory effects of Heritiera fomes, and we've recently discovered 11 phytocompounds that we believe to be associated with its anti-inflammatory usages. Now, chronic inflammation occurs through a number of biological pathways, one of which is the arachidonic acid metabolic network, or the lipid metabolism-associated inflammation, which occurs through lipoxygenase and cyclooxygenase, two major pathways. But in this study, we will be mostly focusing on the lipoxygenase pathway, particularly arachidonic 5-lipoxygenase, or 5-LOX. So in the present study, we wanted to test the hypothesis that these 11 compounds are responsible for the anti-inflammatory properties of Heritiera fomes through regulation of 5-lipoxygenase. And with the help of cheminformatic approaches, we wanted to prove the safety, efficacy, and potency of these compounds as anti-inflammatory drugs. Here is the base outline of the experimental procedures we followed, starting from receptor and ligand information collection and ending with the molecular dynamic simulation and molecular calculation of binding free energies. So first we conducted the in-silico ADMET studies of the compounds and identified which of these fall under the most likely to be drug-like compounds, as well as which ones can cross the blood-brain barrier, in this case methylbetaticin being an excellent candidate, and overall we found tetranurin A to be the best drug-like compound. Then we moved on to our docking procedures. In this case, we validated our docking to cross-docking and redocking. Here you can see the redocked poses of the human arachidonic acid with the native crystal structure. So the docking results reveal apigenin 7p-coumaryl glucoside, tetranurin A, and echinatacin, 3 compounds isolated from Heritiera fomes, to be the best performing compounds when compared to controls. For control, we took 14 drug-like molecules, among which DG051 was the best performer, and we also compared with a natural alternative, which has acetyl-ketoboswellic acid, as well as we take the natural substrate, which is linoleic acid and arachidonic acid, as well as a decoy molecule, which was generated through DUD-E database. Here is the best of conformation of tetranurin A in the 5-lipoxygenase catalytic binding cavity. The remarkable thing here is that while conventions state that most binding energy is contributed through hydrogen bonding, in this case, since the catalytic cavity targets arachidonic acid, which is a fatty acid, the cavity is mostly hydrophobic in nature, so the binding is mostly attributed to multiple pi-alkyl hydrophobic interactions rather than hydrogen bonding. Finally, we wanted to validate these static docking results in a more dynamic platform to better mimic what occurs in the biological systems. For this, we performed molecular dynamic simulations, followed by molecular mechanics calculation of binding free energy through solving the Poisson-Boltzmann equation. As you can see, tetranurin A has the lowest binding free energy among the systems, followed by apigenin 7-p-coumaryl glucoside, and finally the drug compound DG051. And from the trajectories, we can see that both systems stabilize around 60 to 75 nanoseconds, and in case of fluctuations, we can see 2 regions of major fluctuations around 226 and 301 with respect to the APO enzyme. So from these findings, we can conclude that Heritiera fomes as a plant is a reservoir of multiple anti-inflammatory compounds by regulating 5-lipoxygenase, as well as we've identified 2 chemical constituents, tetranurin A and apigenin 7-p-coumaryl glucoside, as candidate drug-like molecules for future anti-inflammatory drug discovery. And last but not least, if you are interested to learn more about Heritiera fomes and its anti-inflammatory activities, here is some previous works from our laboratory, as well as a wonderful review by Violet Hanna regarding the arachidonic acid network. And that's all. Stay home, stay safe. Thank you for your time.